Okay, welcome everyone. Welcome to another episode of our seminar series for the ARC Training Centre in Predictive Breeding. Before we get started, I'd like to acknowledge the traditional owners and their custodianship on the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. To introduce myself, I'm the director of the ARC Training Centre, Professor Lee Hickey, based here at the University of Queensland. And some housekeeping things for today. The seminar runs from 1 o'clock to 2 o'clock. Here in person, we have some tea and coffee and snacks. So feel free to help yourself and continue to enjoy those after the seminar. The seminar is being recorded, so be aware of that. And if you're online, joining us online, and if you have a question, please use the Q&A tab, not the chat function. And if you really like a question in the Q&A tab, you can vote for it and lift it right up to the top. So we ask those popular questions at the end of the seminar. We'll also ask some questions from the floor as well. So think of some really good questions for our speaker today. So it's my pleasure to introduce Dr. Hannah Robinson. She's a quantitative genetics researcher and she has many roles. One of them is an adjunct associate professor role here at the University of Queensland, as well as the Deputy Director of Training and Development with the ARC Training Centre in Predictive Breeding. So it's my pleasure to introduce Hannah. Please welcome Hannah.
All right. Thank you so much, Lee. And thank you everyone for joining both in person and online and giving me the opportunity to talk today about multi-omics predictive modelling and how we are looking at that to accelerate perennial crop improvement. Now, before I get into some of the research that I've been doing at Geisenheim University, I just want to talk a little bit about my journey and how I got to where I am currently in Germany, and as Lee said, wearing many different hats. So I actually started out at university wanting to be a medical doctor. So I was studying pre-med or biomedical science, sat the GAMSAT, did all those things, and was pretty ready to go on and do that. And then wheat and CSIRO and alpha amylase came into my life in a very interesting kind of way. And I ended up actually, doing a summer internship in Canberra at Black Mountain, pretty more in the biochem space, but in wheat and crop improvement. And I think that's where I kind of started to get my love for plant science. I ended up doing an honours with the same group down there in Canberra. And then I moved back home. Brisbane is my home, and did my PhD in barley. So stayed in the crop space, but moved into more genetics. And so I did my PhD with Professor Lee Hickey, and I did it in looking at the genetics of root system architecture for drought adaptation in barley. So did a lot of clear pot glasshouse screening, which some of you may be familiar with, but also some field-based measurements, and then tried to relate that with yield and staging in the field. So some of you may have never seen, oh, that's not it, the pointer, this thing before. That's actually a green seeker. We now have drones. So thank you, technology, for that. And I did just want to point out, during my PhD, I got lots of opportunities here at UQ, and hopefully many of you in the training centre will have similar opportunities to travel. So this is actually me at Akada in Morocco, looking in the field and doing some clear pot screening and teaching the group over there how to do that. So I suppose coming out of my PhD, some of the key skills that I got from that I found really helpful in the next stage of my career was really collaboration, organisation and resilience, which many of you may be able to relate to, as well as some key skills. So for me, since my PhD, I'm continually building on my quantitative genetics knowledge, linear mixed modelling, and then obviously, as you'll see in the next slide, experimental design and multi-environment trial analysis was really, really important to me because I moved directly following my PhD into a role as a junior barley breeder, and that was with Intergrain, who's the leading barley breeding company here in Australia. And I worked in this role not for that long, probably about 4 or 5 years, working under the senior barley breeder, David. And that role was, it was invaluable, really, in setting up my career. So I was responsible from crossing through to probably stage 1, stage 2. We did the handover in the breeding program. And another key role that I had in that process was trying to bring in new technology into the program. So before I started there, we weren't doing any kind of speed breeding, and we're working in inbred. So this became a really valuable tool to rapidly cycle through generations so that we could fix the genetics as quickly as possible before taking those single plants into the field. And I also had a big role in developing the capacity for genotyping in our program. So we co-developed the wheat and barley 40K XT SNP chip with AgVic, and in that process, we also started developing genomic selection models with them, and I had a big role in project managing that, and then trying to implement it into the breeding program as well. So that kind of led in a way to my next role, which was a joint role in a way with Intergrain and with QAFI, the University of Queensland, where I had a role that didn't really have a great name, but kind of like a molecular breeder, in-house quantitative genetics a little bit, and did this across the crops that the company was breeding. A lot of these are just, I won't read them out, but a lot of different roles that I managed in that role, tasks. So a lot of population characterization, trying to understand some of the genetic architecture of our key traits, and we used haplotype-based mapping. Some of you may have worked with me as students where we did that on Intergrain's breeding material. And a big part of that was also looking at how we could improve our genomic selection models for not only selection, but also parental selection as well. And a lot of that again was also spending time going out to conferences and promoting the work we were doing as well. So I finished up this role kind of at the beginning of last year, and then stayed on at Intergrain and in just the Intergrain side of this role until July last year. And then I moved across the world to Europe, and I moved to Geisenheim University, which is kind of central west of Germany, to the Department of Plant Breeding that is led by Professor Kai Vosfels, which some of you may know or had the opportunity to work with. And at Geisenheim, we predominantly focus on horticultural crops, perennials, and in my role there, I, again, wear a number of hats, but recently I was awarded a Marie Curie Post-doc Fellowship, which has enabled me now to focus my work for the next couple of years in the space that I'm going to talk a little bit about, and that is multi-omics predictive modeling. So how can we start layering lots of different omics data together to help improve our predictions, but also our biological understanding of complex traits. So since I've kind of done a big switch from annuals to perennials, I just wanted to, I suppose, highlight some of the unique features of perennials before kind of start talking about a bit of our research. But these crops are really cool. They're highly complex genetically, so they're highly heterozygous. They're typically clonally propagated because we want to maintain and preserve that heterozygosity. They also are regenerative, which means they regrow season after season. And so they experience quite complex environmental interactions over time, which is really super interesting from my perspective. And another key thing from a breeding's perspective, and I suppose from my small time working as a commercial breeder, this just blows my mind. As consumers, we have really strong cultural ties to these like heritage varieties. So if anyone's like a wine drinker in the room or an apple consumer, you probably have your favorite varieties like Sauvignon Blanc from New Zealand or Shiraz from the Barossa, or pink lady apples, all of these things. But a lot of these varieties were bred years and years ago. And so the turnover in the breeding cycle is significantly inhibited by that. And that kind of brings me to some of the key differentials in grapevine breeding, which I'm predominantly going to talk about today. because we have this kind of strong link to these heritage varieties, we kind of do run two different breeding programs. We actually at Geisenheim run a crossbreeding program where we're trying to improve disease resistance by crossing two different wine grapes together. But we also run these clonal selection programs, which you can see a graphic of each of these at the top. And so the clonal selections is basically trying to find this big population of clones, which I'll show you in a second, are phenotypically and genetically diverse, and then selecting within that for your key traits of interest. But in terms of grapevine breeding, no matter which way you go, there's a couple of key features. And the first couple of years is really about getting those F1 seedlings or those clonal cuttings. And yeah, as I said, highly heterozygous, and we're trying to avoid inbreeding depression. After year 2, in this kind of breeding schematic, typically you then need to graft the scion to the rootstock, because in Germany, we have a big issue with phylloxera, so we need the rootstock to be phylloxera resistant. They then go through a hardening phase in the glasshouse and then a transplant into the vineyard into one kind of plot per clone. So anyone that's doing a bit of stats, just keep that in mind as we start working through some of these examples. And so as they're in the vineyard for the first time, they then go into this really long juvenile phase. And this is another thing that was really interesting to me, is that it takes 4 to 5 years for the crop to even develop a berry that's useful in testing. So it really sits in that kind of vegetative phase for so many years. Once it does actually start creating berries, these are the traits around these, which is all we're kind of trying to understand for wine grapes and breeding, are highly impacted by genotype-by-environment interactions. So we have to look at them for a few years. So you're kind of getting to year 10 before you're even going into a multi-environment trial situation. And then again, in multi-trial environment evaluation, you could be doing many, many years of this as well, because you have to, once again, clonally propagate them to get them into all these other environments. So the breeding cycle for wine grapes can be 20 plus years. And this is just phenomenal in my mind. I also want to briefly touch on clonal selection, as it might be a little bit different to some people in the audience. And as I mentioned, we're trying to get a collection of these diverse clones, but they're clones, right? So clones actually accumulate diversity via somatic mutations and epigenetic changes. And this occurs because of that clonal propagation that I was talking about that accumulates over time. And so, as I mentioned, this is really important because of that cultural link to these heritage varieties. So just to exemplify how phenotypically diverse clones can be, this is a sample of our Riesling clones that was collected last year in September. So these are all one variety. Now, I just think this is, yeah, it blows my mind. And this is just a sample, as you'll see in a moment, of what we have at Geisenheim University in the vineyard. In terms of, I suppose, the publication and research in this space around phenotypic clonal variation, there's actually not a lot out there at the moment in Grapevine. So the first kind of thing that Kai and I decided I would work on was really trying to look at the phenotypic variation that we have in a number of different clonal populations, which you can see up here. So we have these 5 varieties, and Riesling was just one of the examples I provided. But you can see we've got really large clonal numbers there. So a lot of the research at the moment in the grapevine space talks about phenotypic variation in clones anywhere from maybe 10 up to max 50. But here at Geisenheim, we're sitting on such a valuable resource of, in some cases, over 1,000 clones of a variety that was bred over 600 years ago. And the reason that we have so many is, I suppose, our the previous professors of our department went out and realized the value of these collections, and went out across Europe, predominantly Germany, France, Luxembourg, and collected these clones from diverse locations. And now they're all situated in our vineyards at Geisenheim University. So our department is just sitting around here as well. So we're very lucky. And as I mentioned, these population numbers are much, much higher than what's currently being published. So just a few very quick examples of some of the variation we're seeing in these populations. This is the raw data for 6 really important traits in grapevine breeding. Of particular importance is degrees Brix, which is basically sugar content, tartaric acid, which is your stable acid component, malic and total, also quite important, and then yield, not as important as what we would consider it in some of our annual crops. But you can see there's pretty significant variation there for clones. And if we drill down a bit deeper and look at it across years and within years, and I suppose I didn't mention, we've got a number of years of data here from 2009 to 2023, and we're continuing to build on this data set. But you can see quite a bit of within and across year variation. This is for yield, and we see similar for Brix and across all the traits. So the next question was, yes, we've got variation there, but how much of that can we actually attribute to the clonal genetic effect? Because again, they are clones. So I fit a pretty basic linear mix model to this raw data, and then produce some BLUPs. I do have a little bit of the code down there. Happy to chat through it later, but there are some limitations to how well I could actually model this. But basically, overall, when we are actually accounting for some of that error variance, we can see that we're still getting some nice variance in our BLUPs. And then if we calculate the broad sense heritability according to the Cullis method, because we have very unreplicated trials, we can see that if you kind of have a look at this dotted circle, this is at the kind of 0.5 mark. Most of the traits for most of the population sit above that. And I think that's pretty impressive, considering, again, we're dealing with a clonal population here. So I'm pretty confident that we can say that it's not only just the year and the field effect driving this phenotypic variance, we've got a pretty strong clone effect. And that's really important because of how slow this process is. And also because we have, so if we have these really a lot of potential in these clonal populations, we have a pretty slow breeding process, and then we have to couple that with, we have a changing environment, and grapevine is very, very much impacted by that. The increase in temperatures has a very big impact on the berry quality. So considering all these things together, I think when we start thinking about some of the new breeding technologies that give us great opportunity to really rapidly increase genetic improvement in grapevine. So as many of us know, we are in a bit of we're accelerating into quite a data-driven discipline that breeding research has become, that has a lot of emphasis on predictive technologies. And I see a lot of value for a number of them being applied at different components of the kind of breeding pipeline that I just outlined. In parallel, we're also seeing quite an advancement in different data sources that are now becoming available due to other technological advances. So typically in the predictive breeding space, we've seen a lot of emphasis on SNP data, but now we're getting more and more technology advances that are making access to some of these other omics data sets more readily available for us as researchers, but I can definitely see a path forward for this in breeding as well, or already is. And so this is where multi-omics predictive breeding really comes into its own. So we see multi-omics predictive breeding predictive modeling, as a really systems-based approach. And so it's really about how we have this kind of really stable foundational kind of genomics layer. And then in between that layer and your end phenotype, you have a lot more flexible regulatory layers that can give you, well, we believe, give you lots more information about about how that genomics layer is then influencing the end phenotype. All of these layers are highly flexible, they interact with each other and also with the environment and the management. So it's a highly complex integrative system, and we see a lot of potential in modeling as many of these omics layers as possible with a number of different trait complexities to then allow potentially improved prediction. But I'm a bit more interested beyond that. Great, let's predict it better, but can we actually use a lot of this additional information to better understand the biology behind a lot of these complex traits, but also interactions as well? So that's where our kind of interest comes from. In terms of the previous research in this space, there's some promising insights in annual crops, which I won't talk about too much today. From what I know, there's been some evidence from maize that they're seeing a significant kind of predictive ability improvement up to 10%, but more importantly, they're starting to see some more interesting underlying biological insights with some of the component traits that are contributing to yield through some of these multi-omics models. So if annuals are anything to go by, there's some promise. The research in perennials is a little bit more emerging, I would say. There's kind of 5 seminal studies to date that have looked at it, and they're generally combining genomics data, so SNP data, with some kind of phenomics data. And I would say in conclusion of all this research so far, it's not as promising as what we're seeing in annuals, but I would say it's highly context dependent, and there's a lot of nuances in these publications. So there's a number of different factors that are influencing kind of the outcomes and things that we need to consider when we are working in this space. I won't go, I've listed a few there, but some of these things will come out as I present some of our kind of initial work. But what the past research is kind of telling us is that there's a lot of space here for perennials for us to do a lot more work to see if there is value in this kind of multi-omics prediction space. And because of that, we're highly interested in it at Geisenheim, and especially in the perennials space. So this is some very preliminary work we've just done on the Riesling clones. So here you can see that beautiful picture again. So the Riesling clones, as I said, we have over 1,000 of them. In this case study, I'm just going to present just over 200. And again, the motivations for this case study is really about how we might develop and apply such a multi-omics framework to this clonal population, and trying to see some of the challenges we may have in this space. So kind of seen this before when we were looking at all of our clonal populations, but what you can see here is just specifically the Riesling data, and you can see again we've got quite a bit of variation within and across here for our 6 traits. For the case study today, I'm just going to focus on yield, and you can notice here as well that it's got a reasonable broad sense heritability of 0.68. So that's the phenotypic variation. In terms of the epi and genomic variation, I'm very fortunate to work with a great bioinformatician, Max Schmidt, at our university, who collaborates a lot with Peter Crisp in his lab. And so they've actually done, they've looked at the genomic variation and found over 1 million SNP mutations in this data set, in this population, sorry. And they've also used a reduced representation library that's been enhanced for unmethylated regions to look at the DNA methylation profiles, and they've found over 500,000 variations there, or the epi alleles. So I think we can confidently kind of say we've got a bit of variation to work with in this case study. So just using that data to kind of characterize the population a little bit, we looked at minor allele frequency, and as anticipated, considering that it's a clonal population, 1/3 of this was relatively rare, which we anticipated, but we also found quite a high minor allele frequency, and we believe this is because of the diverse origins of the clones. And then in terms of the variation that we see in the DNA methylation data, we see a bit of a bimodal distribution again, and we believe this shows that we have some kind of highly conserved methylation patterns as well as some divergent, and again, we believe that's from the origin of the clones. So if we look at this in terms of population structure, just using the SNP data here, we see, and if you focus in on principal component 1 and 2, we basically have very minimal population structure with only 5% of the variance captured by PC1 and 2. again, kind of you would assume this with a clonal population, but it gets really interesting when we have a look at our methylation data. And so looking at principal component 1 and 2 combined, we're getting over 55% of the variation accounted for. And so this kind of suggests that within this methylation data, we're picking up a lot of population structure that wasn't present in the genomic data. So straight away, we're kind of getting some unique information that wasn't present just if we focused on a single omics layer. And so that leads me to what omics data do we have in this kind of multi-omics model? And so post-curation, this is some of the details of the data that we do have. So just over 970,000 SNPs profiling these clones, 450,000 epialleles, representative DNA methylation. And then we also have hyperspectral profiles. So this is kind of our phenomic piece. And we have just under 2,000 wavelengths, and they were captured at a single time point of kind of berry softening. That's a very important point, and we'll come back to that later. And from each of these omics data sources, what we did was we calculated relationship matrices for each of them. We calculated linear variance covariance matrices as well as Gaussian matrices. And I just wanted to also acknowledge my collaborators on this work because capturing all this data is no small feat. As I mentioned, Max and Pete looking after the kind of genomics, epigenomic space. And then I also have to do an acknowledgement to Carlos, who is our physiologist at Geisenheim and does an amazing job. So I've got all of these relationship matrices. And before I want to start modeling them, I really want to have a look at how similar each of them are to each other. For the purposes of comparison, I'm just here showing the linear variance covariance matrices, but straight away from the heat map, you can see that on the top left-hand corner between the genomic and the epigenomic matrix, there's some similarities that visually we can see by eye. There's also a few hotspots that you can see between the GRM and the PRM. But if we have a look at some of the statistics, so one of the most appropriate ways to compare a full matrix to each other in terms of relatedness is through the Mantle test. And here we can see that the GRM and the ERM in terms of similarity are correlated at about 0.39, and this is statistically significant. Between the GRM and the PRM, we're seeing no correlation and no statistical significance. And then for the ERM and the PRM, we're seeing a very, very weak relationship there. If we just have a look at the off-diagonals, so in these relationship matrices, we're really interested in obviously the off-diagonals, the covariance between individuals, and then comparing the similarities of those. We see some slightly inflated numbers for the GRM and the ERM. So we see an R of 0.7. GRM and the PRM, we can see that's now inflated at 0.48. And again, no relationship between our epigenomics and our phenomics. But more interesting, to my interest, I was more interested in the distribution of these relationships. So if you focus in on the x-axis here and just focus on our GRM, we can see that it's pretty narrow and that all of the results have been telling us this. We're not capturing a lot of relatedness between these clones from just our SNP data. But our ERM and our PRM, those similarities values widen from -2 or -4 up to 4. So once again, we're seeing that we're getting unique information from these matrices, but I would caution there's potentially some collinearity there that we may have to deal with in future as well. So what happens when we start multi-kerneling these multi-omics, multi-kerneling these into a multi-omics model? So I've just put up on this screen the different combinations that I've focused on in the results that I'm going to show you there. But basically, it's every kind of combination that you can think. So each individually, then GE, GP, GEP. For the purposes of time today, I'm just going to show you the Gaussian relationship matrices in these multi-kernel models. We did also test the linear forms of the relationship matrices. However, the Gaussian performed better, but the trends were similar. So I'll jump straight into it. And I really first wanted to look at heritability and prediction error variance as my first kind of metric to see, is this worthwhile doing? And again, this is in a linear framework where this is a very simple linear model. We're really just looking at simple genetic effect at this point. And I want you to focus on, well, I'll actually just click through so we can get some lines going. But I just want you to focus on here. These 3 bars are our multi-kernel models where we have more than one omics layer. They're not performing any better than our basically baseline GBLUP GMOD. Similar with the prediction error variances. We are seeing some potential increases with our single PRM and ERM model. And then when we jump to our variance components, so the GRM is in this kind of like olive green, ERM is in this like, yeah, teal, darker teal, and yeah, other teal. But yeah, so you can see that when we multi-kernel these, we're basically losing all effect from the ERM or the PRM in these multi-omics models. And remember, this is a linear framework. And so ultimately what this is probably suggesting is that the GRM is capturing most of the information there and explaining it. And so we're probably really having big issues with collinearity between the GRM and those other omics relationship matrices. So, and in that the other relationship matrices are pushed to the boundary and saying they're not explaining any invariance. So what we wanted to do next was basically remove any component of or any kind of information from these relationship matrices that is highly correlated or explained by the GRM. So basically we wanted to orthogonalize them. And we did it in a pretty standard way where we just basically regressed the GRM effects out of that second kernel model. And we did that individually for the PRM and the ERM. So they're all kerneled based on the GRM. And so these are the orthogonalized models. And straight away, you can see that the ERM looks distinctly different from what it did previously. PRM looks the same, and the statistics tell the same story. So we're seeing now no relationship between the GRM and the ERM. So this suggests that we've successfully orthogonalized it and that they were very collinear. But we're not seeing much difference at all in the PRM, similar statistics again. So this suggests that maybe collinearity is not quite the issue with the phenomics data there. So if we then go back to our multi-kernel modeling, here I am showing the linear relationship matrices models with the orthogonalized side of them, just because, again, when they're orthogonalized and in this kind of linear variance covariance relationship matrices, we're not seeing any difference compared to our standard GBLUP model. And you can see just by them standing alone, they're not contributing much. But if we look at the Gaussian relationship matrices that have been orthogonalized against the GRM for the trait, we can start to see some differences. And this is the part that's pretty exciting for us, that there is an increase in heritability there, or capture of variance for the clonal effect in some of our multi-kerneled models, and there is a reduction in prediction error variance. If we jump to the variance components, and so for that clonal effect, where is that coming from? This is the question we're kind of asking. Is it coming from the GRM or the ERM in these models that we're seeing that increase? And again, focusing on these 3 models on the left-hand side there, you can see really interestingly that the ERM is contributing the most variance. We are getting a slight component coming from the GRM, but overall it's the ERM. A couple of other things I wanted to mention. It would be bad of me not to mention anything about this kind of like PRM or just the phenomic model. And I think there's a lot of work still to do in this space. We are very interested in it. And as I said, we've got an awesome physiologist that I work closely with, and him and I are going to look at this more in depth. But we believe that the challenge with this data set is we just have one growth stage, and we think that we need to longitudinally model that phenomic data. Also, it's highly confounded with the environment and the year and that interaction term there. So we need a bit more complex modeling to get that working. But I suppose the thing we're really interested in this case study is that kind of multi-kernel, multi-omics model. So we're just going to leave that one to the side. Again, to my point, we have a huge amount of variance coming from our random effects here that aren't the clone, and that's our environment, our year, and our interaction of those 2 things together. And so I mentioned before, this is just a simple main genetic effect model. So I haven't modeled any genotype by environment interaction, but I think this just shows that there's a lot of potential in this space for that as well. But overall, for us, we were really excited to see that the epigenomic information is contributing unique and also dominant variants to this multi-kernel model. We obviously wanted to test this a little bit further to see how robust it is in a kind of cross-validation scenario. We came up with probably a very stringent approach, considering, again, the population size, as I mentioned, very small. We're dealing with clones, so very rare alleles in this population as well. All of these things influence your prediction ability. We decided to mask 20% of the population as well as drop 1 year out. So masking both genetics and environment, and sequentially drop that 20% out for each year that we have, so across 11 years, and then do that for 4 other kind of 20% chunks of the population. So pretty stringent, pretty tough. I blame my professor for that one. But I just wanted to, I suppose, give you that background before I go into the 4 key kind of metrics that we're looking at here. Predictive ability, which is your basic correlation between your observed and your predicted. Also bias. So this is where we kind of really want to hit at 0. Anything above, you're typically over predicting. Anything below, you're under predicting. And then really, really importantly, looking at what's happening with the error in our models. And you can notice with both of these, just the general trend of these error metrics, they're pretty much very similar in this data set. But if you did have some outliers for error, you would see a difference between your mean absolute error and your root mean square error. But you can just focus on the RMSE for the results. And basically the red lines here, or the line across your screen, is signifying the baseline GBLUP model. And if you have a look again, focusing on our multi-kernel models that we're most interested in, that the 3 on the end here, that in terms of predictive ability, they are increased above that of our baseline. In terms of bias, they're a bit closer to 0 as well in terms of their average. And, they're on par or slightly reduced in terms of error. So we were we found this to be quite an interesting result, considering the challenges that we had on this prediction scenario. I would also mention here as well, and it's something that's really of interest to me, is I don't really feel that predictive ability or your R, your correlation, is the best way to compare these models. And that's why I'm very interested in bias and root mean squared error. I think because this omics data is capturing error differentially, it's impacting the breeder's equations in different ways. I don't think it's appropriate just to compare error at that very high level. There are some preprints coming out. There's a nice one from UC Davis that's suggesting there's some ways we can correct this. So we'll be looking at doing something similar with these results in the coming weeks. But overall, I think that this is very promising to suggest that there is potential here for these kind of different omics layers to potentially capture more of that clonal variance to increase predictive ability and minimize some of our noise in prediction. So very briefly, and as I said, it's very preliminary results. We wanted to have a look at, okay, so we've got these multi-kernel models, and this is just for our multi-kernel models where we've got the different omics matrices in there. Which of these kernels is driving some of these increases in performance? And we basically backsolved the SNP effects from each kernel in any of the models that were multi-kerneled, and then had a look at their scaled effect against each other. And what you can see here is quite substantially the epigenomic relationship matrix, or the features, the epi features from this matrix, has a higher effect than our SNP or our phenomic information across the top 10 features. And if you actually look at this across the distribution of all the effects. It's really interesting to note that I suppose where you'd see the mean for these epi features is around 5.5 in terms of scaled effects, and we're barely getting to 5.5 in some of these other matrices. So this suggests to us that the epigenetic information is contributing the largest effect. We also had a little look into where, and again, very preliminary, into where some of these epi features may be in the genome. And we found a couple within those top 10 features that are actually sitting in the promoter region of highly conserved genes that may be influencing metabolism and stress tolerance. So this suggests if you do actually find a DNA methylation, like differential methylation in this region, that we are seeing some influence of potentially impacting transcription of a gene in this space, which is super, super interesting. So overall, though, this is just a first look to say that the epigenomic information could be very important in predicting performance in these clones. And so, as I've mentioned, none of this was in any kind of genotype-by-environment interaction modeling. And there's a huge amount of opportunity for this in the perennial space because of how much differences in environment that they do experience over time. And I'm personally just very interested to see if they have a memory of this. And yeah, so many different questions that come out in this space. But we did fit a very preliminary model just to a model that has both the GRM and the EpiRM in it. We basically took each of those omics layers and looked at the interaction of each of those with year here, because we're just dealing with years, or you can think about it like environment. And we fit a factor analytic 2 variance structure to each of those interactions. And then we obviously pulled out the respective kind of yield blobs for those interactions. And you can see that we're getting very, very different kind of signals from each of those. And typically you would probably say that from the genomic relationship matrix, we're seeing a bit more scale genotype by environment interactions. And for the epigenomic data, we're seeing more kind of crossover interactions. And you can also see that the patterns aren't the same across years. So, we're seeing years here that are pretty constricted towards the mean. And then we're seeing the same year in the genomic data that's has a lot more variation. So I think this just demonstrates that there's so much more to do in this space, but that there is some unique contributions to the genotype by environment interaction that we can start to delve into with these omics layers, and that they can be divergent across years. So this can start to suggest that, is there some kind of weather variables that are interacting with these different omics types, and how is that impacting the underlying kind of biology? So in terms of this case study, what did we learn from it? Well, if you're going to look at anything in terms of multi-omics predictive modeling, you really need to consider your collinearity between your omics layers, and that will be different depending on your sources of data. The phenomics needs a lot more resources and requires, as I mentioned, maybe some longitudinal modeling, G/E modeling to really kind of resolve some of those clone versus environmental effects. The multi-omics model did give the best kind of predictive metrics and reduce the noise compared to the genomics, but this is very early. And for us, the epigenomics contributed the strongest effects and highlights some potential value for prediction and clonal improvement. But I did want to kind of come back. Oh, sorry, first before I do that, I did want to say this is early. So obviously, as I mentioned, we want to use this to kind of start to dissect complex traits and that underlying biology of that. Mentioned the phenomics, but again, yeah, the opportunity for G/E for these perennials is really huge in not only leveraging the ability to increase prediction, but also in understanding some of those biological drivers of the interaction. But I wanted to kind of mention here that this is very, very research-based, and being at Geisenheim, we're in the unique position where we're actively running breeding programs for both the scion and the rootstock. And as I mentioned, we've got, crossbreeding and also clonal selection programs. So we have to really think about how can this research be translated into our real world breeding programs. And I just wanted to highlight that there's many, many challenges with this. And that's why, the research to date in perennials has been so context dependent. There's no, at this point in the research, but it potentially will be really important, there's no kind of end-to-end pipeline on how we deal with this highly dimensional complex data that is innately collinear. We have a lot of, you know, biological challenges. To date, I haven't seen a multi-omics model that properly integrates a G/E interaction term and properly models the covariance in that interaction term with the different omics layers. As I mentioned, it's hard to validate these models because of how they capture error differentially, and also practical constraints working on perennials. So we're dealing in small population sizes in generally public breeding programs. So we've got a lot of constraints in that space. And so I just kind of wanted to say, these are the kind of areas, these are the challenges that at Geisenheim we're looking at to work on in the next couple of years with our funding in this space. So really around data integration, how can we best model some of these things? And we believe it goes beyond the linear modeling that I've presented today, and also some of the validation and implementation steps in that space. And yeah, one of the things that we have also been speaking a lot about and starting to propose is, especially for Grapevine, but probably for a number of other perennials, is we really probably need to move to a more collaborative approach, especially if it's still sitting in the public breeding space, following kind of the animal breeding model where we kind of combine training populations and then optimize them for our own prediction population. So we're moving towards that, and obviously using a lot of breeding program simulation and looking to parameterize it with our own variance components to understand where best could this multi-omics technology or parts of it like phenomic selection best fit to accelerate genetic gain or genetic improvement in Grapevine, in both clonal selection as well as in the crossbreeding space. And yeah, so I just wanted to thank all my collaborators on this small piece of work, this case study, as well as Kai Voss-Fels, my professor, and the Geisenheim University Department of Plant Breeding, and our funders. And this is actually a picture of our department, the beautiful Rhine River, and all the grapevines that surrounds us. So I'm pretty lucky to be working in the grapevine vineyards at the moment. So thank you.